6 days after he was born, doctors in Berlin ran an ultrasound on a boy whose thigh and upper arm muscles were already visibly protruding through the soft tissue of a newborn. An image so unusual that it prompted genetic testing before the child had ever lifted anything heavier than his own limbs. By 4.5 years old, he could hold 2 3-kilogram dumbbells extended straight out at his sides, and feat that would challenge many untrained adults. He had no gym membership, no training program, no supplement regimen. What he had was a single mutation, inherited from a mother who had once been a professional sprinter, in a gene most people have never heard of. That gene, and the protein it normally produces, sits at the center of one of the clearest, most direct demonstrations that DNA can shape how a human body builds muscle. And understanding exactly what that gene does and doesn't do, for the overwhelming majority of people who don't carry a mutation this dramatic, is where a much larger and more nuanced scientific story actually begins. The gene responsible for that child's condition is called myostatin, also known by its more technical name, growth differentiation factor 8. Under ordinary circumstances, myostatin functions as a brake, a protein the body produces specifically to limit how much skeletal muscle grows, holding growth in check rather than promoting it. Researchers first identified this function in mice, animals genetically engineered to lack a working copy of the myostatin gene developed muscles roughly double the size of ordinary mice, with individual muscles in some cases weighing 2 to 3 times more than those of unaltered animals. The same pattern turned up independently in cattle. Certain breeds, most famously the Belgian Blue, carry naturally occurring myostatin mutations and are visibly dramatically more muscled than typical cattle as a direct result. The 2004 case published in the New England Journal of Medicine confirmed that the same basic biology holds true in humans. The child carried a mutation disrupting both copies of his myostatin gene, essentially eliminating the brake entirely, and by the time researchers described him in detail, he showed no signs of illness or dysfunction associated with the mutation, simply a body building and holding on to far more muscle than the brake would normally allow. That case is genuinely remarkable. And it's also in the context of trying to understand ordinary human variation in muscle building capacity, something of a red herring if taken too literally. A homozygous loss of function mutation in myostatin, severe enough to essentially disable the gene's protein output entirely, remains exceptionally rare. The Berlin case is still, decades later, cited as one of the most thoroughly documented examples on record. Most people asking why their training partner seems to add muscle more easily than they do are not comparing themselves to someone missing a functional copy of a growth-limiting gene entirely. They're looking at a far more subtle, far more common, and considerably harder to pin down source of variation, one that required a very different kind of research to actually characterize. That research effort has centered for nearly 2 decades on a laboratory at the University of Alabama at Birmingham, led by researcher Marcus Bamman, which set out to answer a deceptively simple question. When a large group of untrained people follow the exact same structured resistance training program, do they actually build muscle at similar rates, or does that assumption fall apart when someone actually measures it precisely? In a foundational study published in 2007 and expanded in 2008, the team put 66 previously untrained men and women through 16 weeks of progressive resistance training targeting the knee extensors, taking muscle biopsies before and after to directly measure how much individual muscle fibers had actually grown, not estimated from strength gains or circumference measurements, but measured at the cellular level. Using a statistical technique called cluster analysis, the researchers didn't assume in advance how many response categories existed. They let the data sort itself, and it sorted into 3 unmistakably distinct groups. A cluster of extreme responders averaged 58% growth in individual muscle fiber size over the 16 weeks. A larger, modest responder cluster averaged 28% growth, real, meaningful, but roughly half as much. And a third cluster, the non-responders, averaged essentially 0 measurable hypertrophy at the fiber level, despite completing the identical training protocol session for session as everyone else in the study. This wasn't a subtle statistical nuance buried in aggregate averages. It was three visibly different biological outcomes produced by the same external stimulus in bodies that had started the study with no measurable difference in baseline muscle fiber size between clusters. The follow-up question was obvious. What specifically was different about the bodies in each cluster before training even began? And what was different about how their tissue responded once training started? The Baman lab's subsequent work traced this down to the cellular level with unusual precision. Skeletal muscle grows in part through the activity of satellite cells, a population of stem-like cells that sit dormant along muscle fibers until activated by mechanical stress, at which point they can proliferate and fuse into existing fibers, donating additional nuclei that help support larger fiber size. The extreme responder cluster, the researchers found, carried a significantly larger baseline population of these satellite cells before training even started, and showed a much more robust satellite cell proliferation and fusion response once training began. The non-responder cluster showed essentially no meaningful increase in satellite cell activity across the entire 16 weeks, despite performing the same mechanically demanding exercise as everyone else. A separate analysis from the same research program pushed the investigation one layer deeper, comparing the pre-training gene expression profiles. Essentially, which genes were switched on or off, and to what degree. In muscle tissue, biopsied, before anyone had done a single training session, the results were striking enough to reframe the entire question. Over 8,000 gene transcripts were found to be differentially expressed between the eventual extreme responders and eventual non-responders measured before training began, meaning these two groups of people, who looked outwardly similar and had comparable baseline muscle size, were walking into the exact same training program with meaningfully different underlying cellular machinery already in place. The extreme responders' tissue showed patterns suggesting the muscle was, in the researchers' own language, already primed to respond to the coming stimulus, with active networks of genes tied to muscle development already engaged. The non-responders' tissue, by contrast, showed elevated markers of pro-inflammatory signaling, a cellular environment that may have actively worked against the kind of repair and growth process training is meant to trigger. This is a genuinely different kind of finding than a single dramatic mutation like the myostatin case, and it's worth being clear about why that distinction matters. The myostatin story is a single gene with a single, well-understood mechanism, producing an extreme, easily visible effect in an extremely rare subset of people. The extreme responder and non-responder clusters, by contrast, appear to emerge from a much more distributed polygenic picture. Thousands of genes contributing small individual effects, adding up to a real, measurable, and fairly dramatic difference in outcome, without any single identifiable gene serving as the clean explanation. That distinction turns out to matter enormously for anyone hoping a simple genetic test might predict their own muscle-building potential, because it means the biology driving most ordinary variation in trainability doesn't reduce neatly to a short list of genetic markers a consumer test could realistically check. That limitation becomes especially clear when looking at what happened when researchers tried the more targeted single-gene approach anyway, searching for specific, individually testable genetic variants that might predict resistance training response, the way myostatin does in its rare extreme cases. The most extensively studied candidate is a gene called ACTN3, which codes for a structural protein found specifically in fast-twitch muscle fibers, the fiber type most associated with power and explosive strength. A common variant of this gene, known as R577X, results in a complete absence of the ACTN3 protein in people who carry two copies of the variant, and that variant has been genuinely, repeatedly associated with elite athletic status, showing up less frequently among sprinters and power athletes than in the general population, and more frequently among endurance specialists. Given that background, the intuitive prediction for a resistance training study would be straightforward. People with functioning ACTN3 protein should respond better to strength training than people without it. A 2005 study testing exactly this hypothesis in women found a result that ran in the opposite direction from what that prediction implied. Women who carried two copies of the non-functional variant, completely lacking ACTN3 protein, actually showed greater relative strength gains after resistance training than women with the fully functional version of the gene. The same study found no significant relationship in the male participants at all. Later studies complicated the picture further still. One trial involving over 100 men found the ACTN3 variant had no relationship with baseline strength or strength gains, but did find that only carriers of the functional version showed measurable increases in muscle thickness specifically. Another study in older adults found yet a different pattern, with women lacking the functional protein actually starting with higher baseline muscle power than those who had it. Researchers who've reviewed this body of work as a whole have been candid about the resulting picture, with more than one analysis concluding that this particular genetic marker may only carry meaningful predictive value in elite athletic populations, and that using it to screen or advise ordinary trainees isn't currently well supported by the evidence. That inconsistency isn't a failure of the individual studies so much as a demonstration of the underlying biological reality the Baman Lab's transcriptomy work had already suggested. Muscle growth in response to training is not the kind of trait that tends to hinge on one or two genes with large, consistent, easily replicated effects. It behaves more like height, or most other complex human traits shaped by evolution across many interacting biological systems, influenced by a very large number of genetic variants, each contributing a small nudge in one direction or another, compounded by satellite cell biology, baseline inflammatory tone, hormonal signaling, and almost certainly factors researchers haven't yet identified. Searching for a single genetic explanation, in other words, was always likely to come up against the same wall the ACTN3 research eventually hit. Real signal buried under enough biological noise that single marker predictions struggle to hold up consistently across different populations and study designs. A separate, considerably larger body of research offers a useful way to check that conclusion from an entirely different angle. Twin studies, which don't look for specific genes at all, but instead estimate how much of the variation in a trait across a population can be attributed to genetics in general by comparing identical twins who share essentially all of their DNA against fraternal twins who share roughly half. A 2017 systematic review and meta-analysis pooling this literature found heritability estimates for muscle strength-related traits, clustering broadly in the range of 50 to 60%. With some individual twin studies estimating heritability for lean muscle mass specifically as high as 80 to 90%. A separate twin study, looking directly at strength training response rather than baseline muscle traits, found statistically significant evidence that how much of a training effect showed up tracked more closely within identical twin pairs than within fraternal twin pairs, for measures including 1 repetition maximum strength gains. A design specifically built to isolate genetic contribution to training responsiveness itself rather than just starting strength. That's a substantial, well-replicated signal, and taken alongside the Baman Lab's cluster analyses, it paints a consistent picture. Genetics clearly accounts for a large share of the variation in how readily different people build muscle. What makes the ACTN3 story, and candidate gene research more broadly, so instructive is what happened when researchers tried to convert that broad heritability signal into something practically useful. A genetic score, built from known variants, that could actually predict who would respond well to training. One study specifically tested this in a large Flemish population, building a composite genetic predisposition score from over 150 known gene variants previously linked to muscle mass or strength in the research literature, then checking how well that score actually predicted real measured outcomes in more than 500 adults. Despite the twin study heritability estimates for these same traits running as high as 66 to 82%, the genetic score built from all currently known contributing variants explained only a small fraction of that variance. A striking, almost paradoxical gap between how much of the trait is known to be genetic in aggregate and how much of that genetic contribution current science can actually attribute to specific, identifiable, testable locations in the genome. That gap has a name in genetics research, sometimes called missing heritability, and it shows up across a wide range of complex human traits, not just muscle-building capacity. It reflects the reality that a trait can be highly heritable in the twin study sense, meaning Genetics clearly matters and clearly varies meaningfully between people while still being driven by such an enormous number of individual variants, each contributing such a tiny effect, that no current test built from a manageable list of known genes can capture more than a small slice of that total genetic signal. Applied to muscle building trainability, this explains something that might otherwise look contradictory. Twin studies confidently establishing that genetics plays a large role, alongside consumer and candidate gene research repeatedly failing to turn that role into a usable predictive test. Both things are true simultaneously, and neither one cancels the other out. None of this erases the reality documented directly at the muscle fiber level in the Baman Labs cluster analyses. The differences between extreme responders, modest responders, and non-responders were real, measured directly in biopsy tissue, and not an artifact of imprecise strength testing or self-reported effort. Genuine, biologically meaningful variation in how readily a given body builds new muscle in response to the same training stimulus clearly exists, and clearly has roots in inherited biology rather than simply differences in effort or technique. What the accumulated research doesn't yet support is the idea that this variation can be usefully predicted ahead of time by testing for a handful of specific genes. The biology driving it appears to be too distributed, too interconnected, and too dependent on factors like pre-existing inflammatory state and satellite cell reserves to reduce cleanly into a genetic report a consumer test could meaningfully interpret. There's a related, somewhat reassuring piece of evidence worth including here because it addresses a question the earlier findings leave open. Does the strength training divergence between extreme and non-responders matter in a practical, functional sense right away, or does it take time to show up? Follow-up analysis of the same research cohort tracked strength gains across the full 16 weeks, not just fiber size, and found an interesting temporal pattern. For roughly the first 8 weeks, all three clusters, extreme, modest, and non-responders, gained strength at a comparable pace, largely because early strength gains in previously untrained people are driven substantially by neural adaptations, the nervous system getting more efficient at recruiting existing muscle fibers, a process that doesn't require new tissue growth at all. It was only in the second half of the study, once the neural adaptations had largely run their course, that the group's strength trajectories diverged sharply. The extreme and modest responders, with their growing muscle tissue, kept adding strength, while the non-responders' strength gains leveled off once the neurally driven early gains were exhausted. That timing detail matters for anyone judging their own progress against a fixed early window. The first couple of months of a new program may say relatively little about a person's longer-term hypertrophy trajectory, since early strength gains are dominated by a mechanism that operates almost independently of the muscle growth variation this entire investigation has been tracing. There's also a practical distinction worth drawing between what non-responder meant inside these specific research protocols and what it might mean for someone's broader potential to build muscle over months or years. The non-responder classification in the Baman studies describes outcomes after 16 weeks of one specific, relatively short training protocol. It doesn't establish that the people in that cluster were permanently incapable of building muscle under any circumstances, only that this particular stimulus over this particular time frame didn't trigger the expected fiber level growth in their tissue. Whether a longer training period, a different loading pattern, or a different combination of volume and recovery might eventually unlock growth in genuinely low-responding individuals remains an open, actively studied question rather than a settled one. The existing research identifies the phenomenon and traces some of its cellular roots without yet fully mapping out whether or how it can be overcome with a different approach. What emerges from putting the myostatin case, the satellite cell research, and the ACTN3 story side by side is a considerably more precise picture than either extreme most casual conversations about muscle building genetics tend to land on. It isn't true that genetics is irrelevant and that identical effort always produces identical results. The cluster analyses make that position difficult to sustain against direct biopsy evidence. It's equally not true that a person's muscle-building fate is written cleanly into one or two genes waiting to be decoded by a cheek swab and a consumer genetics report. the ACTN3 literature's inconsistency, set against thousands of differentially expressed genes found in the Baman Lab's broader transcriptomy work, argues directly against that simpler story. The honest version sitting between those two poles is that real, meaningful, biologically rooted variation in trainability exists, that at least part of it appears traceable to satellite cell reserves and pre-existing inflammatory signaling rather than any single gene. And that for the overwhelming majority of people, everyone outside the rare category of a mutation as extreme as the one identified in that Berlin infant, training still represents the single largest lever available, simply one whose ceiling and pace of return may vary from person to person for reasons science has only begun to fully map.